So that would be Lara Bartels, and so she has a Bachelor of Science, and she's currently at the University of British Columbia.
Okay, hi everyone. Just 1 second, sharing screen. Okay, you should all be able to see my slides. Yeah, as already been said, my name is Lara. I'm at the University of British Columbia, and today I'll be talking about orientation dependence of R2 relaxation in the newborn brain. I'm really excited to be talking about that today because I recently finished my master's thesis on this, and it's also resulted in a publication. The link for that is at the top right, and I hope that by the end of this, you're all very excited to check out this paper. So we'll get right into it, starting with some background. First of all, what is orientation dependence in MRI? So it refers to the fact that in ordered tissues, the signal strengths as well as the relaxation time actually depends on the orientation of tissue microstructure relative to the main magnetic field B0. So orientation of tissue microstructure in terms of, in the example of white matter, for example, just refers to the local direction of a bundle of axons in a white matter fiber. And this is both interesting because we're interested in quantitative mapping of T2 and T2 star in and of itself, as well as that it also affects quantitative imaging methods that are based on measurements of T2 and T2 star, such as myelin water imaging or quantitative susceptibility mapping. Another reason is I think orientation dependence of MRI is interesting in and of itself because most of the mechanisms that have been proposed for orientation dependence are actually deeply linked to the microstructure of the tissue. So by studying how orientation dependence changes, we can also learn something about the microstructure, and usually changes in microstructure predate changes on a larger scale, such as lesions or atrophies. So being able to diagnose early and spot those microstructural changes even before lesions and atrophy become visible would be a great thing to do. I found another few previous works that kind of also highlighted the importance of orientation dependence. One showed that the orientation dependence in R2 actually varies with age, so it's sensitive to that. There was also an interesting study in multiple sclerosis patients that showed that only when they accounted for these orientation effects could they see significant differences between some groups. So it's also an interesting, like a confounding factor that might taking into account might aid with looking at all kinds of quantitative imaging. And then lastly, this interesting paper that looked at cartilage actually and a whole array of different quantitative imaging markers. And they kind of showed that the quantitative parameters that were most sensitive to changes in disease were also most sensitive to orientation dependence. So it's clearly something that affects parameters that we're very interested in and that are highly relevant. So now going on to mechanisms that cause orientation dependence in R2. So in white matter, the most important cause of this is actually down to the white matter microstructure, as I already said, and it's actually the difference in magnetic susceptibility that we've already heard a lot about today between the myelin sheath of the white matter and the surrounding tissue. And we don't only have this susceptibility difference, but as has also been pointed out, the myelin in the white matter is not randomly distributed, but has like an isotropy. So it is oriented or arranged in these, what you can see at the bottom right at the moment, these hollow cylinders that are clearly very anisotropic and have a preferred direction in any given white matter tract. And magnetic susceptibility describes the response of a material to an external magnetic field. And when the material is arranged in such an anisotropic way, the response to the magnetic field also becomes anisotropic. What that means is that depending on whether the axon we're looking at, so one of those cylinders, is arranged in parallel or perpendicular to the external magnetic field, we'll get different effects on the actual magnetic field surrounding it. So when they're arranged in parallel, we pretty much see that the field is very homogeneous around them. So these were calculations done by Wharton and Bartels. And the closer you get, the more you approach a perpendicular orientation, the more inhomogeneous the field surrounding this axon becomes. And when we have field inhomogeneities, we also have accelerated relaxation, and that's how that parameter is affected by orientation dependence. And this combination of, we have magnetic susceptibility differences of myelin in association with this highly anisotropic structure of white matter, then gives rise to this orientation dependence in R2, where relaxation rate is lowest at parallel orientation and highest when we have a lot of inhomogeneities at the perpendicular orientation. And this modeling was suggested by Knight and is actually in really good agreement with the experimental results that have been done so far on relaxation dependent, on orientation dependence of R2 relaxation. And one example is shown here from the work of Bartels from 2021, where they showed that both the myelin water as well as intracellular water, the predictions from this susceptibility-based model agree really well with the observations that they have in vivo. And like we're going to jump a bit away from the brain and looking at one other source of orientation dependency in MRI. And this one occurs in collagen-rich tissues such as tendons and ligaments and cartilage. And there we have a very different functional form of the orientation dependence that is seen on the right, where we have the lowest relaxation rate close to the magic angle of 55 degrees. And also notably, the relaxation rate is lower at high angles compared to lower angles. And in these tissues, the orientation dependency is not attributed to susceptibility differences, but rather that water is found in some kind of structured network. So compared to a sample where the water molecules have all kinds of random orientations, there is a preferred orientation of the water molecules within the tissue. And this means that these dipole-dipole interactions within the water molecules are also happening along a preferred line. And because dipole-dipole interactions are inherently orientation dependent, when the water molecules have an average preferred orientation within a voxel, the R2 relaxation also becomes orientation dependent for this voxel. And this has not been observed for brain tissue in R2 yet. So just to summarize, we saw two different sources of orientation dependence. We have the susceptibility-based model shown in blue here that attributes orientation dependence to susceptibility effects of myelin in the white matter. And the second model I just introduced, that is the model of residual dipolar coupling that is found in collagen-rich tissues. So now, why did we decide to study newborns? As I just reiterated again, one of the key components in the orientation dependence of the adult white matter was this presence of myelin and these susceptibility differences. Now, with newborn white matter, we have the really cool opportunity to study orientation dependence in a human brain that does still have an anisotropic white matter fiber structure. Diffusion imaging shows anisotropy, but the newborn brain is mostly unmyelinated. So we get the chance to study orientation dependency in the presence of an isotropic white matter fiber structure, but the absence of these significant susceptibility differences. And we were just interested in seeing what's going on there. So going on to methods, we looked at both 8 healthy newborn as well as 8 healthy adult subjects. And for orientation dependency of R2, we need to measure 2 things, the orientation and R2. For the orientation, we used diffusion tensor imaging and characterized the local fiber orientation as by the angle theta between the diffusion direction, which we used to approximate the local fiber direction, and the external main magnetic field. And on the right-hand side, you can see some maps of the fiber angles in the newborn and the adult brain. And for our R2 relaxation, we used a gray scan, so gradient and spin echo scan. And to that data, we did a voxel-wise fit of a multi-exponential decay using the decays package. And based on this, we can obtain 2 different compartments, so the myelin water and intracellular water. I think there will be more talks on myelin water on Friday. And for this study, we're going to mostly look at the intracellular water fraction, mostly just because the newborns hardly have any myelin, so there's not really much meaningful analysis to be done there. And on the right-hand side, you can see the R2 maps of the intracellular water, both in newborns as well as in adults. So now the exciting parts, what did we see and what do we think that means? So first, let's look at the R2 orientation dependence in adults again. There we see that our results, both in the intracellular as well as the myelin water, agree nicely with previous findings. And again, the susceptibility-based model that uses the susceptibility of myelin is a really good fit for our data. And now the very exciting part is the R2 orientation dependence in the newborn white matter, which looks distinctly different from what we just saw in the adults. And if we look back at our two models, we can see that it is incompatible with the susceptibility-based model, mostly because we see a higher R2 rate at lower angles, which is opposite to the predictions of the susceptibility-based model. But it is actually in quite good agreement with the model of residual dipolar coupling, which is very exciting because that suggests that we do have some presence of an ordered water network in the white matter. And this means that we probably have some structures there that facilitate this order. This might either be the small amount of myelin that is present, or what we think is a good candidate because of its similarity to the collagen filaments that are present and facilitate the orientation dependency in collagen-rich tissues, are cytoskeletal filaments such as neurofilaments and microtubules, because those are also known to be aligned along the length of the axon. So they also have the spatial anisotropy that is required to be the cause of this order. So to summarize, we looked at R2, the relaxation rate as a function of fiber angle in newborn as well as adult white matter, and our findings in adult white matter were in good agreement with the previous results and best fit by the susceptibility-based model. And in the newborns, in the absence of significant susceptibility differences caused by myelin, we saw that the orientation dependence observed is distinctly different from the one in adults and not compatible with the susceptibility-based model, but instead best fit by a model of residual dipolar coupling. And this is exciting because this effect has not been seen before in R2 relaxation rates in white matter. And it implies that there is some structure in the water networks in white matter, and therefore this orientation effect might also be sensitive to the state of those structures. And the other thing we see, because it's so distinctly different from the adults, we can see that the orientation dependence seems to depend on the myelination of the tissue. So this suggests that orientation dependence is an interesting candidate for a marker for myelination, especially during early development when the orientation dependence is so strikingly different. Yeah, thank you for listening.
Thank you very much, Lara. That was very interesting and very clear talk. So Olivier, do we have any questions?
Yes, we have one question from Christopher Rowley, who asks, since the cytoskeleton structure should still exist in an adult brain, do you suspect that this pattern exists in the adult brain as well, but is being overwhelmed by the effect from myelin?
That is a great question that we've actually spent quite some time thinking about. So I think there is a good chance that this effect is just too small to be seen in the adult brain, whereas that there's just significant amounts of myelin and it's overshadowed. But another thing is that the cytoskeletal filaments are also still developing in the newborn brain. For example, the chemical composition still changes quite a bit over development. For example, I think there's a lot of like phosphorylation going on still, especially in the early times of development. And for the collagen filaments, there a lot of research went into figuring out exactly how they facilitate this alignment of water molecules, and they required quite some distinct free charges that were arranged in the right way. So it might be that chemical changes during development might affect this ability to facilitate structure in water. But I personally think there is a good chance that this is also present in adults, and it is just overshadowed by the susceptibility-based effects.
Thank you, very interesting. Another question from Mustafa Bourara, who asks how to address this orientation dependency in order to recover parameter estimates that are orientation independent.
Yeah, that is also a super interesting question. For a lot of parameters such as R2 and R2 star, those are the ones that I know most about, because I think R1 is significantly less orientation dependent, at least when it comes to white matter. There's some small orientation dependence, but not a lot. But R2 and R2 star, it's quite insignificant. And I think there has been some work, I'm not sure, I think it might be by Lee, who pretty much did that and looked at R2 star and looked at, okay, if we correct for this orientation dependency, that can be modeled usually by some combination of sine squared and sine to the fourth functions. If we take that into account and only look at the, pretty much if you fit an orientation dependent model, you will have some isotropic and some anisotropic R2, and you could look at each of those two individually and map those out. And hopefully just looking at the R2 isotropic should give you a map that is independent of orientation. But yeah, that's a really good question.
Thank you. I think the other questions are, you've pretty much answered them already. So
Perfect. Okay. Thank you.
So thank you again.